Welcome. Welcome to this session of pharmaceutical analysis. Today, we are going to discuss about different pharmaceutical analysis techniques. I'm Dr. Ahmed Nawaz Khan. As discussed earlier about the definition and the scope of pharmaceutical analysis, so just as a recall, the pharmaceutical analysis is a branch of the chemistry which actually involves the series of process for identification, determination, quantification, and purification as well. This pharmaceutical analysis is mainly used for the separation of the component from the mixture, or we can say the analyte from a matrix, and also for the determination of the structure of the compound. As we know that there are different pharmaceutical sources of the pharmaceutical drugs or agents, like plants, microorganisms, minerals, synthetic compounds. So based upon the determination type, these are mainly of two types of the analytical methods. One is the qualitative analysis and second is quantitative analysis. The quality analysis involves the quality or the quality standard of a sample, and identification is also the part of the quality. The quantitative analysis is a method which is used for the determination of the amount of the analyte or the compound in the sample. Now, is there any difference between the method and technique? Yes, obvious. There is a difference between the method and technique. So, analytical technique is a chemical or physico-chemical process. Chemical or physico-chemical process that provide the basis for analytical measurements. We will talk about what are these measurements actually. So, provide the basis of analytical measurements, those processes which are chemical or physico-chemical in nature. So, those processes are known as or described as the techniques. So, then what is the methods? The analytical method is a detailed set of instructions for a particular analysis using a specified technique. So a technique is a chemical plus physico-chemical process that provide the basis for analytical measurements, and a detailed set of the instructions for a particular analysis by using the specified technique, which is maybe a chemical or physico-chemical, is known as the method. So these methods which we are following now, they are already has been validated methods. What is this validated method? The method which was developed and there after that it was validated, it was reconfirmed, it was repeated. So the method which was repeated again and again and reproduced again and again with the same result, that means it get the validated. So those methods which we are following nowadays in our laboratories, in many of our institutions, in the laboratories of the companies, they all are validated method. So when we don't have the method, we have to develop that method. But at this stage, we are just only learning that how we can follow the method. So method is a detailed set of the instructions for analyzing using a specified technique. So now let's discuss about these techniques and some methods. So we have just got to know that what is the difference between the methods and techniques. Technique is a process of chemical and physicochemical nature, and the instruction of following that process is known as the method. So first is the classical method. So to name a few classical methods like the gravimetric method, titrimetric method, and volumetric method. In the gravimetric method, the weight of the sample is determined after the precipitation, and all you know about the precipitation. So the weight of the sample is the basis. So when we take the weight of the sample, then after the precipitation, this is known as the gravimetric analysis. In titrimetric analysis, the volume of the solution is determined after the reaction, such as if the reaction may be neutralization reaction of like the acid and base, it may be a complex formation, it may be a precipitate formation, or it may be redox reaction, which is oxidation and reduction reaction. So the volume of the solution is determined after the reaction, which can be the neutralization reaction, complex formation, precipitation reaction, or the oxidation and reduction reaction. The third classical method is the volumetric method. That is, the volume of the gas evolved by the reaction is determined. Now, this is about the classical methods. There are some instrumental methods also, in which we used instruments, some equipments, just like the electrochemical methods, which are used for the measurement of current, voltage, or resistance. This electrochemical method having a broad area, it is a very broad area, and it comprises of several other techniques or methods, like conductometry, that means measurement of the conductance, potentiometry, measurement of the potential, colorimetry, that means measurement of the current, and voltammetry, measurement of the current at a specified voltage. Now the next method in the instrumental method is the optical method. That means it is based upon the measurement of radiation absorbed or emitted. What radiation? Just like the visible radiation, ultraviolet radiation, infrared radiation. So the absorption method can follow the visible light, can follow the ultraviolet light, can follow the infrared light. So these are the optical methods in which the radiation absorbed or emitted is measured. Next is in instrumental method, next is chromatography. Chromatography, it is the technique which is based on separation of a mixture. The mixture in this chromatography, the in a fluid, it may be a gas, a solvent, or water, that is known as a mobile phase. Actually, this chromatography, when it was discovered, it was based on the color of the some plant pigments. So this is a laboratory technique which is based on the separation of the mixtures. We will discuss on the later stages that what are these chromatography methods. Few examples of these chromatography methods, they are paper chromatography, high pressure liquid chromatography, gas chromatography, right, and the thin layer chromatography and column chromatography, etc. So these are some of the example of the technique which comes under this chromatography. Now the next is methods. Remember, these all methods we are using for the determination of the identification, quantification, and quality. Now, the thermal method is differential thermal analysis, thermogravimetric analysis, and differential element. So, as we are taking the these analytical techniques, and we will discuss them in one by one in the coming session of your pharmacy course. Of which are involved for the determination of a compound: diffractometry, radioactive methods, mass spectrometry, and refractory and polarimetry, etc. So these are some of the basics and very important analytical which we use for the qualitative as well as quantitative purpose. When we choose this, or when we select any analytical method or technique, so there are some factors which affect the analytical method selection. We have a sample and we want to analyze a component, or we want to analyze analyte in the matrix or in the sample. Then what are the factors which affect the analytical method selection? The first factor is the type of analysis, whether it is elemental analysis or molecular or atomic or some other analysis. We should remember that type of analysis we are looking for. We are looking for elemental, looking for some element, we are looking for some compound at the molecular level, or etc. The nature of the material. What is the nature of the material? Whether it is some oxidizing agent, whether it is some reducing agent, whether it is complex forming agent, whether it is acid or base, what is the nature of the material? Plus the sample. That means we are talking about the sample. The third one factor which affect the analytical method selection is the precision and accuracy required. How much precise and accuracy you are required, because we are working on the pharmaceutical analysis, and because some method they have little precise and very less accurate, some method are very precise and very accurate, some are less precise but very accurate, more less accurate. So the requirement of based on the precision and accuracy, we choose the analysis of the sample. The fourth one is the time available for the analysis of a sample. That what is the given time for analysis of that sample? You want to analyze that sample just within 5 minutes, you have to choose a different techniques, a hyphenated techniques, advanced technique. If you want to if you have time for around 3 hours, 4 hours, you can opt some other technique like etc. Fifth is the concentration range of the sample. Is the appropriate concentration we should take while we are analyzing. This is also one of the factors which affect the analytical method selection. Because if we don't have much sample, if we don't have more sample, we have a very less amount of the sample, then again we have to choose appropriate method which can analyze that small amount of sample too. Sixth is availability of the standard for the sample. This is very important, that I'm talking about standard. See, whenever we do the analysis, most of the time we use a standard to which we can compare and say that whether it is right or wrong sample, whether it is identified or not identified, whether the purity is 100% or less purity. So why we are saying, we can obviously, we after analyzing the sample, we can say it is 100% pure. That how can we say it is 100% pure? Because the standard, we use a standard which is 100% pure sample. Based on that, by comparing with that standard, we say that this is the standard, if this is the percentage purity sample. So we need the standard. So the availability of the standard is also very important, and that is one of the factors affecting the applicant selection. And the last is the facilities. Obviously, the feasibility at your work, the facility which are available, all basics, advanced, glassware, chemicals, fumes. You have to look after that whether you have all these facilities or not. That's also one of the factors which affect the analytical method selection. So this is all for today. So see you next time.